From the farm to the laboratory, pigs play a vital role in both agricultural and medical research. Now the genome of the domestic pig has been successfully mapped by an international scientific project which featured contributions from researchers at the Kansas State University College of Veterinary Medicine.
The pig is very important agriculturally, is what a lot of people first think about. And also, though, what for many years, what has been very important about using the pig as a model for human disease. It is, in many respects, better than many of the animal models that we have, depending on the disease, better than the mouse and the rat and some of the other laboratory animals. And so for human medicine, it's been used as a model for obesity, for diabetes, in late onset Parkinson's disease, Alzheimer's disease. And so it's a very valuable animal model for human medicine, and knowing the genome just makes it all that much more valuable because we're able to link some of the traits with specific areas on the genes of this animal.
Over the last 3 years, Drs. Frank Blecka and Yongming Sang contributed data to the International Swine Genome Sequencing Consortium, an organization that has involved researchers from more than 40 institutes in 12 countries. Their labs focused on 2 families of immune-regulating genes called antimicrobial peptides and type 1 interferons, working on identifying and sequencing the immune genes as well as providing genome annotations.
Annotation means you need to define the gene structure, the composition, and after all, you need to define this gene is processing or no processing. After your gene defined, you can comparison with the homologs from other species. But after all, you need to identify the gene's function, like involved in some disease resistance. You need to do experiment to confirm them.
Dr. Sang's and Dr. Bleka's data were combined with results from other labs in the consortium and were published in the November 15, 2012 issue of Nature. The sequenced genome will create opportunities to improve the health of swine herds, such as helping to understand traits that may influence resistance to diseases such as porcine reproductive and respiratory syndrome, or PRRS.
Now, with the completion, at least to this stage where we're out of the swine genome, what this should allow us to do is find out exactly what protein is involved or what mechanisms are involved in these traits. And again, hopefully someday we will be able to have pigs that are resistant to PRRS virus or other disease syndromes that may occur.
Having the completed sequence will further increase the pig's value as a comparative biomedical model for human disease.
So for the pigs, it's the best model for the human because it's physiologically and anatomically it's more similar to humans than like rodents. So the elucidation of this pig's genome sequence, by this way you can compare this trait, this gene, how much this can be represented in humans and how much it is different from humans.
While this is a very monumental benchmark for the swine industry and for swine research, we're not done. There is still much more to learn about the genome, and we have a very good blueprint, if you will, now that will lead us forward, but there's much more to learn in terms of the various proteins that are involved in these traits. While we've accomplished a lot with this recent milestone, I think there's still much more to do.